My name is Terrence Monrian, and today's date is June 7th, 2023. Today, I will be discussing the Week 3 research project Prokaryote versus Eukaryote interaction for the University of New England Online Cell Biology Class 1050. The prokaryote Rickettsia is a small prokaryote which has the characteristics of being obligate, is an intracellular gram-negative bacteria that only reproduces within the host. As you can see, the simple cell structure here is diagrammed out, showing the DNA, ribosomes, the typical plasma membrane and cell wall and capsule of a prokaryote, and the bacterial flagellum, which is used for motility. The Ricketsia's interaction with an animal cell or any other eukaryote is delineated here by 6 potential steps. Step 1, the bacteria enters the eukaryotic cell after being engulfed by endocytosis. Endocytosis allows the bacteria to be engulfed in the membrane and then released into the cytoplasm in a vacuole. Because the prokaryote has the ability to resist phagocytosis and its destruction itself, it is then released into the endoplasm, cytoplasm, where it can then multiply, and here you see multiplication happening in step 3. Step 4 and 6 are 2 possible directions that the cell can go that is reproduced at that point. It can go into adjacent cells through a similar endocytosis, and in 6, we have established a potential symbiotic relationship, which we will talk about in a minute. The other option is it can cause the destruction of the host cell through lysis and be released into the general circulation. The destruction of the host cell is not delineated here, but it is represented here by the cells being on the exterior of the once living cell. According to Lufan and others in a document written in 2020, cross-validation based on a series of trees that were established placed mitochondria robustly within the alpha proteobacteria, which shares a common ancestry with Rickettsia. Endogenosymbiosis is defined as a symbiotic relationship between 2 organisms where one actually lives inside the other. Primary endogenosymbiosis is defined as the original process by which a karyote, such as potentially one of the ancestors to Rickettsia, would become an active part of the eukaryotic cell. While this is still being somewhat approved and believed today, according to Cazzola, Lynn Sagan Mogulis in the 1960s hypothesized this potential about where the mitochondrion came from in the eukaryotic cell. If you look here, I put side-by-side diagrams of Rickettsia and the mitochondrion, and you'll see similarities between the structure. The multi-layered cell with its DNA inside the inner layer with its ribosomes gives the potential for the theory that these are related. If you look at these as being many, many, many millennia of generations between them, and the function and the structure looks very similar. The resources used for this presentation are on these 2 slides. Thank you very much for listening.